Good morning, AP Bio. Miss Kinney here. Today is the first in our series of review videos for the AP Biology exam. In this review video, we'll look at macromolecules and we'll look at the endomembrane system and a little bit on membranes. So the first thing I'd like you to do is at the top of your piece of paper, I'd like you to list all the macromolecules that you know. At this point, I would pause my video. You should list all the macromolecules you know and examples if you can think of any. And then when you restart the video, okay, I'll be writing down my list of examples so you can check your answers against my answers. So to do this, I would start by saying, okay, we have carbs, we have lipids, we have proteins, and we have nucleic acids. And you can see if your list is the same as my list, okay? And after I write my carbs, lipids, proteins, and nucleic acids, I'm thinking to myself, okay, what examples do I know? Specifically, what examples do I know that are used in the cell, either as structure or for energy? So for carbs, I could think about glucose or glycogen or starch, right? And these are all used for energy storage. This is a monosaccharide. Glycogen and starch are polysaccharides, or many monosaccharides glued together. At this point, you might want to think about reviewing hydrolysis reactions and condensation reactions, also known as dehydration synthesis. We also have cellulose, which makes up cell walls in plants. Okay. Then maybe I'd move down to lipids. Okay, and so lipids, we can think about a lot of different lipids. We can think about triglycerides, also known as fats, which are again used for energy storage. And remember, energy is stored between the carbon and hydrogen bonds. So triglycerides tend to store a lot more energy than glycogen or starch just because there's more carbon-hydrogen bonds there. Okay, then we can think about cholesterol, which is found in our cell membranes. We can think about phospholipids, which make up most of our cell membranes. For proteins, there's so many different examples. Just in the cell itself, you can think about like receptor proteins, like the G protein linked receptor, or you can think about enzymes, right? Or you can think about like muscle proteins, like actin or myosin, that help with movement. There are so many protein options, and we'll come back to proteins later. I'll probably have to devote a whole video to proteins and protein folding. At some point, you should review primary, secondary, tertiary, and quaternary structure as well. Nucleic acids, we're talking about DNA and RNA. Okay, so you come up with your list. The next thing I'd like you to do is pause this video, and either on the same sheet of paper or a different sheet of paper, I'd like you to draw a eukaryotic animal cell. Try to draw in as many organelles as you know. Try to be as accurate and specific as possible, and try to label your picture. So at this point, pause the video and do that. And when you're ready, restart and I'll show you my cell. Here's my cell. Okay, it hasn't been labeled yet. So as we're working, you can label yours as well. I drew all of the membranes, both the cell membrane and the endomembrane system, in the same color. So the next thing I think you should do is look at your cell and make sure you've labeled the parts, right? So you have the cell membrane, also known as the plasma membrane. You have the nuclear membrane. You have the nucleolus. You have the ER. You're probably going to have some vesicles floating around. You have the Golgi. Now notice I only drew the organelles in the endomembrane system, okay? Before we move on, I'd actually like you to pause the video again and try to find as many examples of macromolecules as you can in this picture. You might not have drawn out your cell membrane as specifically. I would encourage you to draw your cell membrane specifically before you label the parts. So again, pause the video and try to label as many carbohydrates, proteins, lipids, and nucleic acids as you can in this picture. So when you restart the video, you should have said, okay, here are some phospholipids. And guess what? In the nuclear membrane, there are phospholipids. The ER is also an internal membrane made up of phospholipids. The Golgi is made up of a phospholipid bilayer. Vesicles are tiny phospholipid bilayers. So across all of these organelles in the endomembrane system, you'll find phospholipids, which are part of the lipid category of macromolecules. And that's why this is called the endomembrane system, because it's all made up of the same phospholipid bilayer that the cell membrane is made up of. We think about the endosymbiotic theory, when larger prokaryotes engulfed or ate smaller prokaryotes, which became the mitochondria and the chloroplast, and then they used their own membrane to fold it internally to make this endomembrane system, the inside membrane. Okay, so I have some lipids labeled, but remember, cholesterol is also a lipid that helps maintain fluidity in the cell membrane, right? The tails of the phospholipid bilayer are bent, and this cholesterol helps prevent the phospholipid bilayer from freezing together. We also have proteins, right? This could be an intermembrane protein, or some sort of internal membrane. Maybe it's an enzyme. Maybe this is like a G protein linked receptor, and this is the G protein. And maybe once it's activated, it's going to activate this enzyme, adenyl cyclase. So cell signaling goes along with everything we've learned about so far. So I've labeled proteins, I've labeled lipids, okay? Maybe this isn't a G protein linked receptor. Maybe it's just an MHC protein like in the immune system. So we can add a carb group to it for recognition. Nucleic acids, right? Where do we find nucleic acids? Well, we find the DNA in here. And if we're doing transcription, you're also going to find mRNA. So at this point, you've labeled a bunch of different macromolecules inside this structure. Okay, next I want you to pause the video and I want you to draw the path of a protein through the endomembrane system. What would that look like? Okay, so take a second, pause the video. Where's the protein made? Where could it go, etc. Okay, so hopefully you've taken the time to do that. As we remember in transcription, the DNA, a section of the DNA, a gene, is transcribed into mRNA, right? You have processing, and you're going to get your shortened mRNA when the introns are cut out, okay? This mRNA can go two places. It can either attach to a ribosome on the ER, or it can attach to a free-floating ribosome. mRNA that's attached to free-floating ribosomes tends to make proteins that are used in the cytoplasm. Okay. mRNA that attaches to a ribosome on the ER tends to make proteins that are going to travel through the endomembrane system. So a couple things can happen in the ER, right? This protein's going to fold from primary structure, which is the chain of amino acids, to secondary structure, where you'll get the alpha helices and beta plated sheets, to the tertiary structure, and maybe to quaternary structure, where two or more polypeptides have joined together. So that's part of what's happening in the ER, this protein folding. We can also modify this protein. We could give it a carbohydrate tail here. So now we have a glycoprotein. Next, this protein is going to be transported via vesicles to the Golgi. Okay? And this vesicle just buds off the ER because the vesicle is made up of phospholipid bilayer just as the ER is. So a vesicle really just is a part of the ER that's broken up, and it's going to travel via the cytoskeleton and those cute little motor proteins that I like to the Golgi. In the Golgi, that protein could be modified further. Part of the protein could be cut out to give it its mature form. It gets a zip code, so it tells you where it's going to go. So let's say this is a protein that we want to put on the cell membrane. So we're going to give it the cell membrane a zip code 22003. This protein would get a tag much like a zip code 22003, and then it would be stuck on the membrane. Right? It would be transported via vesicle to that membrane as well. Okay, so that's one way that this protein could go. A different protein could maybe flow to the lysosome via vesicles. And again, the lysosome is made up of a phospholipid bilayer, and it contains digestive enzymes to break down old food, right? It could be taken, this protein could be taken for export out of the cell. So when we think about like the pancreatic cells making insulin or glucagon, that protein could be exported. That's the power of the endomembrane system. You can really help modify a protein and you can transport it where you want it to go. Okay, at this point to finish off the video, I just want you to add some other things to the cell. So please add a mitochondria and a chloroplast, and as you're adding them, just think about their membranes as well. So pause the video, add them, and then check back in with me. So here's my mitochondria. I made a mistake earlier in the video. I said add a chloroplast, but if this is an animal cell, it does not have a chloroplast. So we're just adding our mitochondria. So the mitochondria has an outer membrane made up of phospholipids. It has an inner membrane made up of phospholipids. It has the matrix. It has the inner membrane space. Okay? And all of this form relates to its function. The matrix is full of enzymes that help do the Krebs cycle. Right? The inner membrane space allows the proton gradient to build up, so you have a proton motivating force to help you make ATP. Embedded in the inner membrane among the phospholipids are proteins, right, that help with the electron transport chain. So the mitochondria is an amazing structure because form follows function, right? The matrix is a liquid form that contains enzymes that help with the Krebs cycle. The inner membrane is highly folded to pack as many of these proteins in as possible so you can have a lot of electron transport chains. Those electron transport chains take the protons from NADH and FADH and pump them into the inner membrane space. The inner membrane space is very narrow, and so you can build up the proton gradient quickly, and then you have the ATP synthase that these protons can flow through, causing the ATP synthase to spin so that ATP can create it. And finally, oxygen takes those protons coming through and the electrons, and water is formed. Okay, but also you make a ton of ATP. Okay, and again, they think the mitochondria came into the cell via the endosymbiotic theory. So that's review video 1. I hope you enjoyed it, and we'll see you soon for another review video.